I want you to start off today by picturing something for me.
Okay.
Just take a second, wherever you are, and picture a peaceful meadow.
Oh, that's nice.
Right. Maybe there is a gentle breeze rustling through the tall grass. You've got a few wildflowers dotting the landscape.
Maybe a couple of butterflies drifting by.
Exactly. It is quiet. It is tranquil. I mean, it is the absolute picture of serenity.
It is a very comforting image. It is the kind of landscape that, lowers the human heart rate just by looking at it.
Which is incredibly ironic, because I am about to completely shatter that illusion for you.
Oh, here we go.
Yeah. If you were to look at that exact same meadow through the sensors of a chemical ecologist, you would not see peace and quiet.
Not at all.
You would see a roaring, chaotic, unrelenting battlefield. The air itself would be thick with these invisible, desperate SOS signals.
That's intense.
You'd see inescapable molecular fingerprints left behind at the scene of microscopic crimes. And you would be standing right in the middle of evolutionary arms races that have been escalating for hundreds of millions of years.
It's true. Switching the lens from the acoustic and visual spectrums that we evolved to process over to the chemical spectrum, it reveals a constant, deafening broadcast of information.
A completely different world.
Exactly. Every single plant in that meadow is locked in a high-stakes dialogue. They are communicating with the organisms trying to consume them and the organisms that might actually offer them salvation.
And today, we are wading directly into that invisible war zone.
Gonna be fun.
We are looking at an incredible dossier of landmark studies that really map out the chemical ecology of plant and animal signaling.
And we are covering a lot of ground today.
We really are. We are tracking a trail of research spanning from these foundational breakthroughs in the 1980s all the way to massive global meta-analyses published in the 2020s.
Some really cutting-edge stuff.
But as we comb through this data, I mean, from the microscopic intricacies of caterpillar regurgitant to the subcontinental synchrony of entire forests dropping seeds, we have to tread very carefully.
We do.
Because popular science loves to run wild with the idea of plants, like talking or plotting against us.
Or having feelings.
Right. So our mission here today in this deep dive is to aggressively distinguish between established, rock-solid molecular mechanisms and the newer, heavily contested hypotheses that still have, quite frankly, massive evidence gaps.
We are separating the textbook science from the flashy headlines.
Exactly. Okay, let's unpack this. What are the rules of engagement here?
Well, that distinction you just mentioned is paramount, particularly because the human brain is just this relentless pattern recognition machine.
We want everything to be a story.
Right. When we observe complex, highly effective strategies in nature, our immediate biological instinct is to assign intent.
Yeah.
We want to see a mind at work.
We want there to be a mastermind.
Exactly. But the absolute foundational rule for our exploration today is that we must ruthlessly discard the metaphor of conscious plant intent.
No plotting plants.
None. Plants do not have nervous systems, they do not harbor desires, and they definitely don't map out cognitive strategies.
Right, so there is no botanical war room where the oak trees and the lima beans are huddled around a map, moving little insect figurines with a stick.
That is a hilarious image, but no. We are looking entirely at the outcomes of natural selection.
Okay.
When a plant executes a chemical defense that seems brilliantly strategic, it is the result of blind, unthinking survival over deep time.
Deep time.
Yes. Millions of years ago, random genetic mutations caused a specific biochemical response. The plants that possessed that mutation survived at a slightly higher rate than those that did not.
And so they lived to pass on that trait.
Exactly. This relentless evolutionary math slowly carves out behaviors that perfectly, almost uncannily, mimic intelligence.
It's just survivor bias.
Purely. Synchrony, chemical mimicry, targeted volatile release. These are the weapons, but natural selection is the general.
Keep that framework in mind because we are going to test it immediately. Let's dive right into the micro-level dynamics of a single leaf under siege.
Let's do it.
There is a concept in chemical ecology known as the bodyguard hypothesis. Have you ever heard of this? It centers on the phenomenon of indirect defense.
Which is fascinating.
It is. Instead of a plant fighting off an attacker itself, say, by growing thicker thorns or synthesizing a fatal toxin, it outsources the violence.
It calls for backup.
Exactly. And the foundational work on this goes back to the 1980s with Marcel Dick's research on the lima bean, Phaseolus lunatus.
A classic study.
Dick was observing what happens when these beings are attacked by two spotted spider mites.
Which, for context, is a very common, highly destructive agricultural pest.
They just wreck crops.
They do. And when those mites begin puncturing the plant cells and draining the contents, the lima bean initiates this profound metabolic shift.
It flips a switch.
It does. It begins synthesizing and releasing a highly specific blend of volatile organic compounds into the surrounding air.
And here's the crazy part. Those compounds aren't meant for the spider mite.
No, they aren't.
They are an olfactory homing beacon for a predatory mite, Phytocoelus persimilis, which is an arachnid that specifically hunts and devours the two-spotted spider mite.
The plant is under attack, and it summons this specialized killer of its attacker.
Marcel Dick's work was instrumental in building the, what he called the infochemical framework to describe this.
Right, because the plant is bridging trophic levels.
Let's break that down for our student listeners.
Sure. The primary producer, the plant, is entirely bypassing the second trophic level, which is the herbivore. It skips right over it to directly recruit the third trophic level, the carnivore.
It was like calling the cops instead of fighting the burglar.
That's a great way to put it. But it is crucial to note the mechanics of this relationship.
Yeah.
The plant is not offering a direct nutritional reward to the predator.
It's not paying the bodyguard.
Exactly. It is merely advertising an opportunity. It is chemically broadcasting the presence of prey.
It is basically throwing up a neon diner sign that says, you know, fresh spider mites right here.
Yeah.
And while the lima bean study laid the groundwork, the sheer drama of this mechanism wasn't really fully appreciated until a landmark 1990 science paper by Ted Turlings, Jim Tumlinson, and Joe Lewis.
Oh, this paper is legendary.
They moved the focus away from beans and mites over to maize seedlings and caterpillars, specifically beet armyworm caterpillars munching on corn.
A very different system.
But just like the lima beans, the chewed corn releases a specific volatile bouquet that summons a third party.
Right.
But in this case, the third party is a Cotesia parasitoid wasp.
And we really need to emphasize the distinction between a standard predator and a parasitoid. It's vital for understanding why this is such an efficient evolutionary strategy for the plant.
Because it's not just a quick meal.
No, a standard predator might eat a caterpillar and leave. A parasitoid wasp relies on the living body of the caterpillar to incubate its young.
It is a phenomenal, terrifying life cycle. I mean, the wasp flies into the volatile plume generated by the corn. It locates the feeding caterpillar, and it uses a highly adapted ovipositor to inject its eggs directly into the living tissue of the insect.
It's straight out of a sci-fi movie.
It really is.
The wasp larvae hatch,
Yeah.
and they begin consuming the caterpillar from the inside
Oh,
out.
yikes.
And here is the incredible part. They selectively feed on non-vital tissues first. They specifically avoid the central nervous system and the major organs to keep the caterpillar alive.
It's keeping the meat fresh.
Exactly, preserving their fresh food source.
Mm.
But crucially for the corn plant, the parasitized caterpillar almost immediately ceases its normal feeding behavior.
Oh.
The internal trauma and the physiological hijacking by the wasp larva stop the caterpillar from doing any further significant damage to the host plant.
So the plant calls in an airstrike and the threat is neutralized.
Essentially, yeah.
But what elevates this Turlings, Tumlinson, and Lewis work from just a cool observation to rigorous textbook science is their control group.
Yes. The control group is everything.
Because I could argue, right, that anytime you tear a plant leaf, it leaks chemicals.
Yep.
Like if you mow your lawn, you get that powerful, fresh-cut grass smell.
Right, which is just a massive passive release of green leaf volatiles due to structural damage.
Exactly. So they had to prove the corn wasn't just, you know, bleeding passively.
That is the crux of the experiment. They utilized mechanical wounding to isolate the variable.
How did they do that?
They took razor blades and physically excised tissue from the maize leaves, perfectly mimicking the area and the mechanical disruption of a caterpillar bite.
Wow, just cutting tiny little caterpillar bites by hand.
Tedious work. But they discovered that this purely mechanical damage did not trigger the complex, wasp-attracting volatile bouquet.
So the razor blade wasn't enough.
No. The plant required the actual biological presence of the feeding caterpillar to initiate the full defensive cascade.
The physiological implications there are just staggering. I mean, the plant possesses the sensory architecture to differentiate between abiotic damage like a razor blade or a falling branch or strong wind, and a biological assault by a specific herbivore.
It knows what is attacking it.
And the sophistication of that sensory architecture was expanded upon a few years later in a 1995 follow-up study published in PNAS.
Right, where they investigated the spatial extent of this volatile release.
They wanted to know how far the signal travels.
Exactly. If a caterpillar bites a single leaf on the lower quadrant of a maize seedling, they found that the localized damage initiates a systemic alert.
Systemic meaning the whole body.
Yes. Eventually, completely undamaged leaves on the upper portions of that same plant also begin synthesizing and emitting the parasitoid-attracting volatiles.
That systemic response, it reminds me of a commercial fire suppression system in a high-rise building.
Oh, that's a good comparison.
Right. If a fire breaks out in a server room on the second floor, a localized alarm doesn't just ring in that single room.
Of course not.
The sensory input, the heat or the smoke, triggers an electrical cascade through the building's central wiring. Suddenly, strobe lights are flashing and alarms are blaring on the 30th floor, initiating evacuation protocols everywhere simultaneously.
That is a highly accurate mechanistic analogy. In the plant's case, the wiring involves electrical signals, hydraulic pressure changes, and internal signaling hormones like jasmonic acid traveling through the vascular tissue.
Through the xylem and the phloem.
Yes. The initial, highly localized bite triggers a biochemical cascade that propagates throughout the entire organism. It is a fully active, meticulously regulated, whole-body defensive program.
Which naturally forces us to ask the ultimate mechanistic question.
Right.
If this entire systemic wasp summoning program is lying dormant in the corn plant, what is the exact biochemical key that unlocks it?
That's the million-dollar question.
Because if the razor blade didn't work, how does the plant cellularly detect the caterpillar?
Unlocking that mystery required an absolute masterclass in chemical sleuthing.
I can only imagine.
The researchers knew the trigger had to be something transferred from the caterpillar to the plant during the act of feeding.
Some kind of physical exchange.
Right. And this led to a breakthrough 1997 science paper by Hans Alborn alongside Turlings and Tumlinson. They focused entirely on the caterpillar's regurgitant.
Which is just a fancy word for spit.
Yes, the oral secretions constantly bathing the wound as the insect feeds.
So they spent years isolating every single microscopic compound in that caterpillar's spit.
It was a monumental effort.
They would separate a fraction of the spit, apply it to a mechanically wounded leaf, and just wait to see if the plant released the wasp-attracting volatiles.
It is an unimaginably tedious process of elimination.
Until they finally isolated a single, highly active compound.
And they named it volicitin.
Volicitin. The isolation of volicitin was a watershed moment in chemical ecology, right?
Absolutely. To confirm their findings, they took a purified synthetic version of volicitin, they applied it to a fresh razor blade cut on a maize leaf, and they observed the plant mount the exact same systemic volatile response as if it were being consumed by a live beet armyworm.
Boom.
Volicitin was the definitive biological on-switch.
But when we look at the molecular structure of volicitin, the evolutionary narrative gets incredibly intense.
It gets very complicated.
Because volicitin is a fatty acid amino acid conjugate, or a FAC. This molecule isn't something the caterpillar simply generates from scratch.
No, it's not.
It is a molecular chimera.
Yes, the biochemistry here is fascinating for anyone studying this. The fatty acid portion of the conjugate, specifically linolenic acid, is actually appropriated from the plant itself.
The bug steals it.
It does. When the caterpillar breaks open plant cells, it ingests these plant lipids. Then, in the highly alkaline environment of the caterpillar's midgut, those plant fatty acids are chemically bound to an amino acid glutamine.
And the glutamine comes from where?
The glutamine is synthesized by the insect's own metabolic processes. The fusion of the plant's stolen lipid and the insect's amino acid creates volicitin.
Okay, looking at it from an evolutionary arms race perspective, this seems like a massive tactical error by the caterpillar.
Why do you say that?
Well, why would it synthesize a bespoke molecule that functions as a homing beacon for a wasp that wants to lay eggs inside its brain?
That is the logical question. It seems paradoxical until you understand the constraints of the caterpillar's physiology.
Okay, lay it on me.
These fatty acid amino acid conjugates are absolutely critical for the insect's survival. They function as powerful surfactants.
Like soap.
Essentially biological detergents, yes, inside the caterpillar's digestive tract. They facilitate the breakdown and assimilation of plant proteins. The caterpillar cannot digest its food without synthesizing these conjugates.
I love conceptualizing this. Here's where it gets really interesting. I view this as a burglar breaking into a house.
Okay, let's hear the analogy.
The burglar doesn't bring their own tools. They find a crowbar in the homeowner's garage.
Right, that is the plant's fatty acid.
Exactly. But to get a good grip on the crowbar, the burglar uses their own custom monogrammed leather gloves.
And that is the insect's glutamine.
Right. And as they pry open the safe, the glove gets permanently fused to the crowbar and left at the scene. The insect literally cannot extract the nutrients from the plant without leaving this highly specific, inescapable molecular fingerprint inside the wound.
That perfectly illustrates the inescapable nature of the interaction. These molecules, these unavoidable metabolic byproducts left at the scene, form the basis of a broader category known as HAMPs.
Herbivore-associated molecular patterns.
Exactly. The plant relies on these HAMPs to reliably differentiate between a passing breeze and a lethal threat.
And returning to our foundational rule about evolution, the plant didn't actively figure this out. It didn't decide to scan the caterpillar's saliva for surfactants.
Not at all. Over millions of generations, plants were constantly subjected to random genetic mutations. Eventually, a mutation arose that caused certain plant surface receptors to bind with this specific ubiquitous caterpillar byproduct.
It was just an accident.
Pure chance. And another subsequent mutation linked that receptor activation to the jasmonic acid signaling pathway, which, as we discussed, controls volatile production.
Right.
Because those specific plants could suddenly recruit third trophic level bodyguards, they suffered less herbivory, they dedicated more metabolic energy to producing seeds rather than replacing eaten leaves, they dominated the gene pool.
It is the blind filter of natural selection forging a lock and a key out of the chaos of digestion and cellular mutation.
Beautifully put.
It is a flawless textbook example of molecular ecology. We have the behavioral outcome, the systemic response, and the exact isolated molecular password.
It's all there.
But that level of clarity is a luxury in science, because as we pivot to the next study in our dossier, we are stepping out of the well-lit laboratory of insect biochemistry and into a much murkier, vastly more complex arena.
We are scaling up from the relatively simple digestive tube of an insect to the massive, convoluted microbial bioreactor that is a mammalian digestive system.
Mammals! This brings us to a 2017 study published in Functional Ecology by Bettina Ohse and her team.
A very heavily debated study.
They were investigating whether the password in the spit concept applies to roe deer browsing on wild maple and beech saplings.
Right.
The premise is compelling, right? If a tree can recognize the spit of a bug, can it recognize the spit of a 50-pound mammal?
The field ecology required to test this is notoriously difficult. You cannot precisely control the feeding behavior of a wild ruminant to isolate variables.
You can't just ask a deer to take one specific bite of one specific leaf.
Exactly. So Ohse's team had to rely on a rigorous simulation.
Right. They couldn't just unleash deer into a greenhouse and take precise molecular measurements. They simulated the browsing by taking clippers and physically severing the buds and shoots of the sapling.
Mimicking the damage.
But the critical variable they introduced was a pipette. Immediately after severing the shoot, they pipetted genuine roe deer saliva directly onto the fresh, exposed vascular tissue of the cut.
And of course, they established a control group of saplings that experienced the exact same mechanical clipping, but received no saliva application.
The mammalian equivalent of the razor blade versus the caterpillar spit.
Precisely.
And the data they extracted was incredibly provocative. The saplings that received the salivary treatment exhibited distinct, rapid shifts in their internal chemistry compared to the mechanically clipped controls.
Real measurable changes.
The researchers tracked a surge in phytohormones, particularly salicylic acid, which is a foundational signaling molecule in plant defense pathways.
Very different from the jasmonic acid used for the insect pests.
Furthermore, they observed a significant upregulation in the production of defensive secondary metabolites, specifically tannins.
Tannins are polyphenolic compounds that are highly effective against mammalian herbivores.
How do they work?
They bind to salivary proteins and digestive enzymes in the animal's gut. It essentially renders the plant tissue highly astringent, bitter, and fundamentally difficult to extract nutrition from.
Like eating an unripe persimmon.
Exactly like that.
So when this paper dropped, the science communication machine just went into overdrive. The headlines were totally definitive. Trees recognize roe deer by their saliva.
They were everywhere.
It felt like we had found the mammalian volicitin. The narrative was perfect. But this is exactly where our mission today, to distinguish between verified mechanisms and contested hypotheses, becomes critical.
Yes, we have to pump the brakes here.
Looking closely at this study, the leap from the data to the headline feels enormous. First off, dripping saliva from a pipette onto a clean scissor cut completely strips away the physical reality of a deer taking a bite.
It really does.
A deer forcefully tears, crushes, and splinters the wood. It introduces massive structural torque. Doesn't that fundamentally alter the plant's stress response compared to a surgical cut?
Your skepticism is entirely warranted, and it highlights the inherent limitations of simulated field ecology. The mechanical stress of tearing versus cutting creates vastly different wound topographies.
Which matters.
It matters greatly because it can drastically alter how quickly and deeply saliva penetrates the vascular tissue. But the evidence gaps go far deeper than just the physical application.
Okay, let's hear them.
We have to look at the phylogenetic breadth of the response. Ohse's team tested exactly 2 tree species, maple and beech.
Just 2.
And the data showed that the chemical response was highly species-dependent. The maple and the beech did not deploy identical defensive cascades in the presence of the saliva.
So we cannot universally claim that trees recognize deer. We can only say that these 2 specific species exhibited a localized reaction.
Precisely. But the most glaring, monumental gap in this research, and the reason we must categorize this as an active hypothesis rather than a textbook law, is that we lack the molecule.
The volicitin.
Right. With the caterpillars, all born isolated, we know the exact molecular key. With the deer saliva, the scientific community has absolutely no idea what the active volicitor actually is.
We know the saliva is triggering a response, but we are completely blind to the actual mechanism.
We are in the dark.
Is it possible there is the mammalian version of a ham pea? Like the specific protein in the deer's saliva acting like volicitin?
It is possible, but isolating it is a biochemical nightmare. Consider the complexity. An insect's regurgitant is relatively straightforward. A deer is a ruminant.
Oh, right. They chew cud.
Yes. Its saliva is an oceanic soup of thousands of dynamically fluctuating proteins, digestive enzymes, shed epithelial cells, and a wildly diverse microbiome.
So many variables.
Are the trees reacting to a specific mammalian salivary enzyme? Or are their receptors actually detecting the molecular signature of a specific strain of bacteria living on the deer's teeth?
That's a great point.
Furthermore, a deer's saliva contains highly variable concentrations of growth hormones. The tree might not be detecting deer at all. It might simply be reacting to an influx of foreign hormones.
The signal-to-noise ratio in that saliva is just overwhelming.
Exactly.
So how do we correctly categorize this research? We obviously don't just throw it out. The data regarding the spike in salicylic acid in tannins is real peer-reviewed science.
We categorize it as a profound ecological clue. The Ohse study conclusively demonstrates that plant defense systems are exquisitely sensitive to the complex chemical mixtures present in mammalian saliva.
Right.
But until the specific molecular elicitor is isolated, synthesized, and proven to independently trigger the pathway, the assertion that trees recognize deer remains a compelling, unverified hypothesis. It is science operating at the raw frontier.
I love that. It shows that the textbook isn't closed.
Never.
So far, we have examined the chemical ecology of plants reacting to the immediate trauma of being eaten. The animal acts as the trigger, the plant acts as the responder.
Right.
But for our next source, we are shifting the paradigm entirely.
Oh, we are flipping the script.
We are moving from reactive defense to proactive, landscape-scale reproductive ecology. We are going to explore how forests effectively weaponize their own seeds against the animals that eat them.
If we connect this to the bigger picture, we are transitioning from the microscopic scale of molecular elicitors to the macroecology of continent-wide population dynamics.
We are talking about mast seeding, or masting.
A phenomenal event.
If you live near an oak forest, a beech forest, or certain coniferous ranges, you have likely witnessed this. Masting is a phenomenon of extreme synchronized reproductive volatility.
Volatility is the perfect word.
For several consecutive years, an entire forest might produce almost 0 seeds. We call these the bust years. The trees are photosynthesizing, they are growing leaves, expanding their root networks, but they are hoarding their carbon and completely withholding reproductive effort.
Building up energy.
Then suddenly, in a single synchronized season across thousands of square kilometers, every tree erupts.
The boom year.
They initiate a massive coordinated boom year, dropping an unfathomable ground-covering glut of acorns or pine nuts all at once.
The sheer biological energy required for a forest to transition from a bust year into a full masting event is staggering. It requires a massive internal reallocation of resources.
And the evolutionary rationale behind this extreme boom and bust cycle has been a central question in ecology for decades.
It has. But a major 2022 global meta-analysis published in PNAS solidified the leading explanation for this behavior.
Which is the predator satiation hypothesis.
Yes, it is essentially an evolutionary numbers game played out over generations.
The primary adversaries in this scenario are the local seed predators, the squirrels, the jays, mice, and wild boars that rely heavily on the high-fat, high-protein content of those seeds to survive the winter.
Right. And if an oak tree simply produced a reliable, moderate crop of acorns every single year, the local squirrel population would effortlessly adapt to that carrying capacity.
They would just eat all of them.
They would consume nearly 100% of the seed crop, leaving virtually nothing to germinate. From the tree's perspective, steady reproduction is an evolutionary dead end. It is just feeding the enemy.
I view masting almost like a biological distributed denial of service or DDoS attack.
I love this.
In computer science, a DDoS attack overwhelms a server by flooding it with so much simultaneous traffic that the server's processing capacity completely crashes, allowing the attacker's true payload to slip through the chaos unnoticed.
That is a phenomenal conceptual framework. During a masting boom year, the forest is executing a biological DDoS attack against the collective digestive capacity of the local seed predators.
Flooding the servers.
Exactly. By coordinating a synchronized release of billions of seeds across the landscape, they flood the biological processors. The local squirrel and jay populations gorge themselves. They cache as much as they possibly can.
But they physically cannot consume or hide the sheer volume of biomass dropped on them.
They are completely satiated.
And the leftover traffic, the surplus acorns that the crashed servers couldn't process, are the ones that survive to germinate and become the next generation of oaks. The sheer volume ensures survival.
Yes.
But the DDoS attack only works if the servers are kept vulnerable, which brings us to the crucial importance of the bust years.
The bust years are the setup for the eventual attack. If the forest masted every year, the predator populations would exponentially explode to match the infinite food supply.
You'd just have a billion squirrels.
Right. By following a boom year with a long, unpredictable stretch of near-zero seed production, the trees systematically starve the seed predators.
It's brutal.
It is. The carrying capacity of the forest plummets, causing massive die-offs in the rodent and bird populations.
So by the time the trees have accumulated enough carbon reserves to launch the next synchronized boom year, the predator population is at its absolute lowest, weakest point,
Guaranteeing maximum seed survival.
The logistics of that synchrony are what break my brain. How do millions of individual trees, separated by varying elevations, topographies, and microclimates, all execute the boom year at the exact same time?
It's a huge puzzle.
Because they don't have a shared biological intranet to coordinate the drop.
No, they require a universal environmental cue that cuts across all those localized microvariables. And a fascinating 2024 study, published in Nature Plants, provided a profound piece of that puzzle.
What did they find?
The researchers analyzed decades of masting data across subcontinental scales and linked the synchronization trigger to a fixed astronomical event, the summer solstice.
The longest day of the year.
Yes. The research suggests that the trees are utilizing a combination of daylight duration sensed by specialized light-sensitive proteins called phytochromes and the accumulation of specific temperature thresholds surrounding the window of the solstice.
So it's both light and heat.
Exactly. Because the solstice occurs universally on the exact same day, regardless of whether a tree is in a valley or on a ridge, it provides an unalterable celestial metronome.
Wow.
If the temperature parameters align perfectly with the light cues of the solstice, it triggers a system-wide hormonal cascade that redirects carbon away from vegetative growth and forces massive floral and seed production.
Okay, here is where I have to step in and check our language.
Good idea.
Because even as we describe this, it is so tempting to slip back into anthropomorphism. When we talk about forests systematically starving rodents and executing coordinated DDoS attacks based on a celestial metronome, it sounds like we are describing a highly organized botanical syndicate.
It does. It sounds incredibly intentional.
And that is the exact cognitive trap we identified at the beginning of our deep dive. The metaphor is a useful shorthand for ecologists to describe the devastating effectiveness of the system, but it is a catastrophic failure if we apply it to the actual biological process.
We must return to the cold math of natural selection.
So walk us through the evolutionary math. How does a subcontinental DDoS attack evolve without a single tree ever knowing what a squirrel is?
Imagine an ancestral forest millions of years ago. The trees in this forest lack synchrony. They drop their seeds
The
randomly.
free-for-all.
Right. Some trees drop heavy loads one year, neighboring trees drop heavy loads the next. The localized seed predators act as a highly efficient mobile cleanup crew.
They just walk to the next tree.
Exactly. They simply migrate from the heavily producing trees to the next, consuming everything. The trees utilizing asynchronous reproduction experience a near-total failure to recruit new saplings into the understory. They are evolutionarily sterile.
But genetic mutation is a constant engine of variation.
Exactly. Purely by chance, a random genetic mutation occurs in a small population of trees, linking their reproductive hormonal pathways to the specific temperature and light cues surrounding the summer solstice.
An accidental connection.
Yes. Because these mutated trees share the exact same environmental trigger, they accidentally synchronize their seed drives. For the very first time in that forest's history, a localized glut of seeds occurs that slightly exceeds the immediate digestive capacity of the local predators.
And a few of those seeds survive.
And those surviving seeds carry the solstice trigger mutation.
Over thousands of generations, the environmental pressure of seed predation acts as a relentless filter.
The asynchronous trees continually fail to reproduce. The synchronous trees continually manage to overwhelm the predators and leave offspring.
So the synchrony spreads across the landscape, not through communication, but through pure survivor bias.
That is exactly it. The trees aren't coordinating an attack, they are simply reacting uniformly to a universal trigger, because every single ancestor that failed to react to that trigger was eaten out of the gene pool.
That reframing is profound. The synchrony isn't a plot. It is a statistical inheritance carved out by starvation and survival. It perfectly mimics strategic intelligence, but it is entirely blind.
Is the essence of evolution.
Now, masting relies on overwhelming the animals that want to destroy the seeds. But for our final exploration today, we are going to look at the inverse relationship.
A completely different strategy.
We are going to examine an evolutionary strategy where the tree entirely abandons the DDoS approach and instead completely surrenders its reproductive future to the very animal eating its seeds.
We are moving into the realm of scatter-hoarding mutualism. It is a system built on profound, lifelong biological dependency.
When animals like squirrels or jays cache seeds during a masting event, they are burying them at optimal depths in aerated soil to eat later.
Right.
But caching relies on spatial memory, and memory is fallible. Animals forget caches, or just as often, the animal falls prey to a hawk or a fox before winter ends.
And those unrecovered caches are effectively planted forests.
The animal acts as an unwitting silviculturist. And the most extreme, highly specialized example of this dynamic is detailed in Diana Tombach's seminal 1982 research regarding the Clark's nutcracker and the white bark pine.
Nucifraga columbiana and Pinus albicollis. If you want to witness the absolute extremes of coevolutionary pressure, you look at high-elevation ecosystems.
It's a harsh world up there.
It is. The whitebark pine dominates the harsh, unforgiving subalpine zones of the western United States and Canada. The environmental constraints there are so severe that the tree has abandoned typical methods of seed dispersal and forged an absolute reliance on the Clark's nutcracker.
The physical anatomy of the white bark pine cone is the ultimate proof of this dependency. If you picture a standard pine cone, you picture scales that open up as the cone dries, right? Allowing the wind to catch the winged seeds and carry them away.
But the whitebark pine has evolved an indecent cone.
Indehiscent, meaning it doesn't open.
Correct. Even when fully mature and dried, the scales of the cone remain sealed tight, locking the seeds inside. Furthermore, the seeds themselves have entirely lost their aerodynamic wings.
Because they don't need them.
Exactly. They are large, dense, and packed with high-value lipids and proteins. If the Clark's nutcracker does not arrive to physically dismantle that cone, the seeds will remain trapped inside until they rot on the branch. The tree is completely incapable of independent reproduction.
It has locked its entire genetic future in a biological vault, and only one specific species of bird possesses the key.
And the bird has evolved incredible physiological adaptations to serve as that key. The Clark's nutcracker possesses a long, sharply pointed, specialized bill engineered specifically to pry apart those stubbornly closed scales.
It's like a custom tool.
It is. And even more remarkably, it has evolved a highly specialized sublingual pouch,
A pouch under its tongue.
an expandable sac located under the floor of its mouth, specifically designed for bulk transport.
It acts as an internal biological cargo hold. A single bird can extract dozens of these dense, heavy seeds, pack them tightly into its sublingual pouch, and fly several kilometers across rugged topography.
Far away from the parent tree.
It will then land and systematically bury these seeds in small caches across the landscape. And the scale of this operation is staggering. Our sources note that a single Clark's nutcracker can cache upwards of 30,000 seeds in a single season.
30,000.
Hmm.
The bird relies on its phenomenal spatial memory to recover the majority of those seeds throughout the deep alpine winter, sustaining itself when all other food sources are buried under feet of snow.
But it always caches a massive surplus as an evolutionary insurance policy.
Exactly. The thousands of seeds it abandons or forgets are perfectly positioned for germination. The mutualism is complete. The bird is guaranteed a high-energy winter food source, and the tree guarantees its offspring are dispersed far beyond the shade of the parent canopy into prime establishment sites.
It is an evolutionary masterpiece, but it is also an incredibly fragile bargain.
It really is.
The deeper the dependency, the higher the risk of catastrophic failure. And our sources highlight an incredibly ominous conservation crisis unfolding right now. The white bark pine is facing unprecedented compounding threats across its entire range.
The species is being decimated by a dual onslaught. First, an invasive fungal pathogen known as white pine blister rust, to which the native trees have almost no evolutionary resistance.
It just wipes them out.
And second, massive, unprecedented outbreaks of native mountain pine beetles, which have been supercharged by warming winter temperatures that fail to kill off the beetle larvae.
Whole mountainsides, entire stands of these ancient, ghost-like subalpine trees are rapidly turning red and dying. And because the trees are dying, the overall density of cone production across the landscape is crashing.
This is where the threshold dynamics of mutualism become terrifying. The Clark's nutcracker is a highly mobile, highly intelligent forager. It constantly assesses the energy landscape.
Doing the math.
Yes. If the density of healthy cone-producing whitebark pines in a specific watershed drops below a critical threshold, the energetic cost of flying to that stand outweighs the caloric reward of the remaining seeds.
The birds will simply abandon the dying forest and seek out alternative food sources elsewhere.
Exactly. And when the birds abandon the stand, the ultimate tragedy of the indehiscent cone is realized. There may be a few healthy, blister rust-resistant whitebark pines remaining in that forest.
Trees that hold the vital genetic key to the species' survival.
But without the nutcracker to pry open their cones and transport their seeds, their genetic legacy is trapped. The mutualism collapses. The survival of the bird and the continuation of the tree are evolutionarily fused. If the bird decides the trip isn't worth the calories, the forest is functionally extinct long before the last tree falls.
It forces a profound realization. We love to draw these neat taxonomic boundaries in biology. The plant kingdom is over here in this textbook. The animal kingdom is over there in that textbook.
It's not like that.
When you drill down into the chemical signals, the reproductive strategies, and the mutualistic dependencies, you realize those boundaries are illusions. The communication, the survival, the very continuation of the landscape, it runs inextricably straight through the gut and the memory of the animals.
The plant shapes the physical reality of the animal, and the animal dictates the genetic future of the plant. It is a singular, unbroken loop of selective pressure.
So, to synthesize the incredible journey we have been on today, we started down at the microscopic level, unpacking how a plant can mount an active, systemic defense, literally summoning third trophic level wasp bodyguards when it senses the specific biological trauma of an attack.
We examine the inescapable chemical betrayal of digestion, seeing how caterpillars are forced to leave behind custom molecular fingerprints like volicitin that plants have evolved to detect.
We waded into the skeptical, messy frontiers of real-time field ecology with the mystery of mammalian saliva, exercising our critical thinking to differentiate between verified molecular pathways and exciting but unproven hypotheses.
Scaled up to the landscape-level evolutionary math of masting forests, where millions of trees synchronize their reproductive effort to the summer solstice, executing a biological DOS attack that brilliantly mimics strategic intelligence, but is fundamentally driven by the blind power of natural selection.
And finally, we examined the delicate, lifelong mutualism of a bird planting a high-altitude pine forest from memory, and the cascading conservation tragedies that loom when those intricate evolutionary bargains are threatened.
If there is one overarching principle to extract from this dossier of sources, it is a deep respect for the staggering, unthinking power of natural selection. It is a process devoid of intent or foresight. Yet, operating over deep time, it engineers relationships so intricate, biochemistry so precise, and ecological cycles so tightly wound that they appear indistinguishable from conscious design. We are observing biology functioning at its absolute highest level of complexity.
It really is a marvel. Thank you for walking us through the mechanics of that complexity and for helping us strip away the metaphors to see the actual science underneath. It makes the reality of the forest far more impressive than the fiction.
It was my pleasure.
And for you listening, thank you for joining us on this deep dive into the invisible world. Before we sign off, I want to leave you with one final provocative concept to mull over.
Oh, this is a good one.
Something that builds directly on the molecular and ecological pathways we just explored. We talked about how natural selection spent millions of years forging these hyper-specific locks and keys.
The plant receptor that perfectly fits the caterpillar's unique spit.
Exactly. Or the hormonal pathway that perfectly links the light of the summer solstice to the optimal temperature for seed production.
Very finely tuned systems.
So what happens to this entire silent, ancient language when human-driven climate change alters the environmental variables faster than evolution can possibly rewrite the genetic code?
That is the crisis we are facing.
Think about the masting forests. The summer solstice is a fixed astronomical reality. The tilt of the Earth isn't changing. But the temperature thresholds that have historically accompanied that day are shifting drastically. What happens to the synchronization of a billion oak trees when the celestial calendar says one thing but the climate says another?
Does the biological DDoS attack fail?
Or consider the arrival of novel invasive species. What happens when a completely foreign insect, transported across the globe by human trade, begins chewing on a native plant? An insect that doesn't produce volicitin. An insect that doesn't speak the local chemical dialect.
A total breakdown of communication.
Will the plant recognize the damage as biological, or will it mistake it for the wind? Will it know to call for its bodyguards, or will the forest, in the face of these rapid, unprecedented changes, suddenly find itself deaf and mute?
It is a sobering thought.
Think back to that peaceful meadow we asked you to picture at the very beginning of this deep dive. The tranquil grass, the quiet breeze. The next time you walk through a forest, or even just observe a potted plant on your windowsill, remember that silence is a complete illusion.
It is never quiet.
There is a deafening, desperate, highly complex conversation happening right under our noses, shaped by millions of years of survival. Keep questioning that invisible world around you. Until next time, take care.